All right, so today we're going to go over some basic math skills that are required to be successful on the AP Biology exam in this course. And these are the types of math things that I wouldn't necessarily teach through the scope of the course, but they will show up on tests and can show up in the AP Bio exam because they're kind of general math skills that we assume that you can kind of do, but it trips you up because you sometimes, you're seeing it out of context when you've learned these skills previously. So some types of things that we're going to look at: doing rate calculations using slope, how efficient a process is, an efficiency calculation, how many times greater something is than something else, what percentage of something is something, like all that stuff are things that they could require you to do in the AP Bio exam. So let's try an example first of calculating rate. So the first thing you're going to do is we're going to calculate slope using the y equals mx plus b formula, or rise over run, right? So let's look at question 1. Use the graph above to calculate the mean rate of population growth, individuals per day, between day 3 and day 5. So the way you would do this is you take your change in y, which in this case would be from day 3 to day 5. So we find those points here. Here's day 3. Here's day 5. So we'll call day 5 about 880, let's say. So day 5 is 880. And we'll subtract that from what day 3 is, which is 200. And then we're going to divide that by the difference in the number of days, which is 5 minus 3, which is 2. So if you do that math, you get 480. You get 680 divided by 2, which is equal to 340 in that case. Okay, so 340 individuals per day. For this one, a little bit harder, though a similar type of formula. You can use the formula in a similar way, but it requires you to look at a specific line. So it says, based on the data shown, calculate the average rate of increase in oxygen consumption for the animals acclimated to 5 degrees. So we're going to look at the line with the circles on it, not the triangles, as it increases from 10 to 30. So 10 to 30, that's going to be our change in y, or change in x. So we're going to put 20 in the denominator. And then the numerator, we're going to do that difference, which in this case looks about, again, 180 approximately minus 80. That ends up giving you 100 divided by 20. And if you do that, you get 5. Now here's where it gets a little confusing or complicated. It says to the nearest tenth. So if you just calculated 5, that is not correct. It's 5.0 in that case, because it says to the nearest tenth. You actually get no points or no credit if you do not put 5.0, even if you do it correctly. All right, so those are rate calculations. Let's talk about energy efficiency calculations. So I don't know if you know that, but like most processes, when you're transferring energy from one form to another, are not going to be 100% efficient. The amount of energy you put in is not going to be what you end up getting out, and that goes to kind of the next thing. For example, like automobiles are only about 10% efficient. What that tells you is the gasoline you put in your car, only about 10% of it is used to actually move your car. The rest of it is lost as heat and is waste energy. So it's a fairly inefficient process, actually. Now, if we look at this, I actually did the calculation for you here at the bottom. The transfer of energy from glucose, which we use for energy, to ATP, which is our internal energy currency, our cellular energy currency, is about 33.9% efficient. That's a fairly efficient process compared. It's only a third of it's transferred, but it's still way more than the 10% in an automobile. So the way we calculated this was there's 690 kilocalories per mole. That's just a measurement of the energy per mole of glucose, and about 233.6 kilocalories per mole that ends up in ATP from burning that 1 mole of glucose. So as you can see, it's 233.6 divided by 690, only about 33.9% efficient. There's an additional calculation here because on the actual question, you're going to get this later in the year, by the way, so if you know how to do this now, you're going to have an easy time later. 7.3 kilocalories per mole of ATP, so each ATP molecule is 7.3 kilocalories per mole, and 32 ATP are created through cellular respiration. So we multiply 32 times 7.3, that's where we get 233.6. Then we did the efficiency calculation, 233.6 divided by 690 times 100 to to get 33.9% efficient. Okay. How many times greater is one thing than another? This is a tougher one because there's a lot of words in here. I'm going to show you how you kind of find what you want to find. So what we did was, in this case, the graph below, I'll just read from here. Don't worry about all of the rest of it. It's about an enzyme and converting one molecule to another. The graph below shows that at certain concentrations, ATP inhibits the enzyme, whereas AMP activates it. According to the information presented in the graph, when the concentration of fructose 6-phosphate is 0.5 millimoles, so its concentration of fructose 6-phosphate is 0.5 millimoles. So we're looking at this value right here, right? 1, 2, 1.5, 2.5, etc. We then go and we look here at let's see, express your answer to the nearest whole number. Okay, how many times more active is PFK in cells with 1 millimole ATP plus 0.1 millimole AMP? So that would be this one with the circles. How many times greater is that than the 5, which is the black circle right here? So we look at that value, we go up to both lines at that point, and you can see here this one's 5 and this one's 0.5. We take 5 divided by 0.5, we get about 10 times greater in that case, okay? So that's how you do a question like that. Some other graph interpretations. We have, so based on the data in figure 1, identify the concentration of gossypol that reduced C. parvum growth to 50% of that in control trials. This one's a pretty easy one. You go to 50% here, 0.5 or 50%, and then we go to the line, we go down, and that's approximately 15, maybe 16 micromoles of gossypol concentration. Okay, so that one should be, as I said, 15, 16. 124 is part of a study to observe the activity of photosynthetic organisms in a temperate zone lake. Measurements of light penetration were made at different water depths. The results are presented in the graph below. Note that higher light penetrating values indicate a greater amount of light reaching the water at that depth. According to the data above, what's the deepest water level at which light penetration is expected to be at least 200 micro, I don't know, whatever this measurement is, and, at that. So we go to 200, we go over to the line, we go down. So now it says to the nearest tenth, so this is about a half, right? So this is 5, each of these worth 1, so about 1.5, whatever this meters is going to be your answer. So that's the deepest water at 1.5 that you're going to have that much light reach that point. 1.5 to the nearest tenth. If it said to the nearest hundredth, you'd have to say 1.50. All right, this one was a tough one from what I remember. When this came out, I thought, oh man, this is brutal. So what they're saying is this is the percent activity, all right, of an enzyme, let's say, ATPase activity. It's actually a pump that moves ions back and forth, so it's not an enzyme. It uses energy, ATP. This is 100% efficient. We go down, and this is looking at, okay, this is 30% efficient, 20% efficient, so on and so forth. The scientists determined that the ATPase activity in PKD cells treated with 1 picomolar, so right here, is 150 units. So they're telling you it's this arbitrary 150, let's say, ATP hydrolyzed per second at concentration of 1, which is representative of 100% approximately activity. Well, what's it going to be when it's at 10 to the 6th? So we go to 10 to the 6th, notice it's 30%. So it should be, it could be a proportion, or you would just say, what's 30% of 150, right? And if you do that calculation, 30% of 150 is 45. So 45 units of ATP hydrolyzed per second. That's how you do that one. And the last calculation we're going to do is, it deals with like the law of conservation of matter and energy, the first law of thermodynamics. So we have this energy accumulated in biomass is 1.9 times 10 to the 4th kilojoules per mole, or kilojoules per meter squared. Of this biomass, there are 3 layers: the tree layer, the shrub layer, and the herb layer. So what you're going to do here is you're going to say that all this has to add up to this. The only thing that makes it a little bit more confusing are these are all in scientific notation. What you can do is you can actually convert them out of scientific notation if you want to. So it's like 19,000 is the total equals 13,000 plus x plus 1100, right? Because 1.1 times 10 to the third is 1100. You then solve for x. And I think this one ends up being, let me do this calculation. I have to do it on my phone real quick. Let me see. So I said it was 13,000 plus 1100 equals 14,100, and we're going to subtract the original 19,000 minus 14. Did I do that right? Yep. 14,100, and you end up getting 400 or 4,900 for that in that trouble over, or 4.9 times 10 to the third. Now, it says the nearest whole number, so you can just say 4,900 and you would get credit for that. Okay, that was the math calculation part of this first unit. If you have any questions, come see me.